Hi everyone, welcome back to the lecture series on overview and integration of cellular metabolism. We have reached week 4 and we are continuing with carbohydrate metabolism. Today's class is on galactose metabolism and associated disorders, and the concepts that we will be covering in today's class are the pathway of galactose metabolism, the biochemical abnormalities in galactosemia, the clinical manifestation of galactosemia, the diagnosis of galactosemia and management of galactosemia, that is galactose metabolism and its associated disorders. Okay. So, right off the bat you need to know glucose. I have already discussed glucose and galactose are monosaccharides during digestion absorption of carbohydrates. Now, in that class you see, if you count the number of carbon atoms, both of them have got 6 carbon atoms. So, 1, 2, 3, 4, 5 and 6. If you count the number of carbon atoms, the formula is simple, C6H12O6. We have all read in our from school days, right? This is the formula of glucose, but this is also the formula of galactose. So, glucose and galactose are isomers, right? Specifically, glucose and galactose differ in the 4th carbon, where the H and OH is actually reversed. If you have followed closely my HMP shunt lecture 1, over there I told you the isomer in which the HOH is rotated in any carbon other than the penultimate carbons, other than the terminal carbons, so leaving apart 1 and 6, if in any 2, 3, 4, if in any of these carbons H and OH are rotated, then this type of isomerism is known as, were you able to answer? Epimers. Okay, so glucose and galactose are epimers, E-P-I-M-E-R-S, epimer. Okay. So, with this information let us go forward. So, again glucose is a monosaccharide and the major source of glucose is lactose, major source of galactose, sorry, is lactose. Okay. So, lactose is a disaccharide that is composed of glucose and galactose, and the primary source is milk and milk product, that is dairy products. So, who breaks down this lactose into glucose? That is lactase. Again, during the class on digestion absorption of carbohydrate, I told you the deficiency of lactase enzyme leads to a disease known as lactose intolerance, right. So, suppose we do not have lactose intolerance, lactase is intact and now the glucose and galactose are being absorbed by the intestinal system, right. So, lactase is hydrolyzing lactose into glucose and galactose and then they are absorbed. How galactose is produced in our system? The absorption is about the oral food intake. It is produced from lysosomal degradation of glycoproteins and glycolipids. It is one of the process by which galactose can be produced in the body, but the major source is from diet, especially milk and dairy product. Where galactose is metabolized, very important multiple MCQ question, galactose is metabolized almost exclusively by liver. Okay. So much so that galactose tolerance test is one of the functional indicator of liver capacity, right. Next fundamental ground thing that we need to know, whenever we need galactose in any reaction, okay, galactose cannot directly participate. It prefers to be donated, handheld by someone, and who is the donor of galactose during synthetic reaction? That is UDP glucose, uridine diphosphate. So, galactose is bound to UDP, it is in the form of UDP galactose where galactose is donated. This concept is not new to you, you have already seen it in previous lectures of glycogen synthesis, right. UDP glucose is donating glucose for glycogen synthesis, same way UDP galactose is donating galactose for any synthetic reaction. So, considering now we have got galactose in our system, the main problem why we need to study galactose metabolism is what happens to this galactose molecule, how it is metabolized, what are the reaction or changes or enzymes that are acting on galactose to produce the clinical features, right. So, the first reaction is actually galactokinase. From your knowledge previously, I am sure you have encountered this kinase group of enzymes, hexokinase, glucokinase, phosphofructokinase, all of them are doing what? They are adding a phosphate molecule or phosphate group. So, kinase adds a phosphate group, and galactokinase is also not an exception. So, upon action of galactokinase, galactose is converted to galactose 1 phosphate, right. This is the first reaction, and this is the cyclical form of galactose. Again I am telling you, if you are having difficulty in memorizing the structures, be sure you just memorize the metabolic pathway by the name of substrates and products. So, galactose is being converted to galactose 1 phosphate, very important. Glucose by the action of hexokinase or glucokinase is being converted to glucose 6 phosphate. Galactokinase converts galactose to galactose 1 phosphate. The phosphorylation occurs in the first carbon, right. In the next step, what happens? Well, the most important enzyme of galactose metabolism, I told you, galactose 1 phosphate uridyl transferase. Since there is UDP glucose, UDP galactose, that is donating galactose, we need synthesis of UDP galactose. So, how UDP galactose will be synthesized? Remember I told you galactose cannot directly participate in any reaction. If we need formation of, suppose we need formation of lactose, I am just giving you a very small example. We need glucose and galactose. So, galactose cannot simply go and bind to glucose, simply like glycogen. So, we need galactose in UDP form. So, how this UDP galactose is formed? Again with the help of this enzyme galactose 1 phosphate uridyl transferase, where galactose 1 phosphate and UDP glucose is combining to form UDP galactose. Okay, this is the reaction. Galactose 1 phosphate and UDP glucose under the action of the enzyme galactose 1 phosphate uridyl transferase, often abbreviated as GALT, is getting converted to UDP glucose, right. UDP galactose, sorry. So, once UDP galactose is formed, okay, see galactose 1 phosphate reacts with UDP glucose to form UDP galactose and glucose 1 phosphate, right. Glucose 1 phosphate, mind it, often this product or this thing is spelt wrongly, okay, glucose 1 phosphate in the presence of galactose 1 phosphate uridyl transferase. So, what happens next? So, up to this I hope it is clear. So, there are two things that you need to keep in mind. UDP galactose is the active donor of galactose in any synthetic reaction. So, what synthetic reaction? UDP galactose is essential for the formation of compounds like lactose, glycosaminoglycan, glycoproteins, cerebrosides, glycolipids, etc. These are the compounds that needs galactose, and galactose needs to be donated in the form of UDP galactose, fine. In the next step, what happens? UDP galactose can be converted back to UDP glucose. Okay, so when we need galactose for synthesis, UDP galactose can be formed and it can donate galactose as much as it wants to. But when the requirement of galactose is gone, the excess UDP galactose can be formed or converted back to UDP glucose by the enzyme. You tell me, what is the basic structural difference between UDP glucose and UDP galactose? UDP is same, glucose and galactose is also same, only glucose and galactose are different, and glucose and galactose are itself epimers. So, in the looking at the structure of whole UDP galactose, if we just locate that 4th carbon and tweak the HOH, it will become UDP glucose, right. And you are absolutely right in guessing, UDP glucose and UDP galactose are epimers. So, the enzyme is galactose 4 epimerase, okay, UDP galactose 4 epimerase. So, this enzyme then acts on UDP galactose and leads to formation of UDP glucose. So, now what happens? Galactose is channeled to the metabolism of glucose. So, this galactose, these are optional, see galactose 1 phosphate or see glucose 1 phosphate, this one. What is the use of this glucose 1 phosphate? This glucose 1 phosphate can be converted to glucose 6 phosphate by the enzyme phosphoglucomutase, very important. Again a special class of enzymes that helps in transfer of a molecule intramolecularly, transfer of functional group intramolecularly. So, glucose 1 phosphate becomes glucose 6 phosphate. So, if we look at the structure of glucose, the phosphate group is being shifted from first position to the sixth carbon. Why? Because now glucose 6 phosphate is essential for all other carbohydrate metabolism reaction. So, glycolysis, TCA cycle, HMP shunt, everything, right. So, galactose in this way can take part in glucose metabolism. Thus, galactose can be channeled into, you see. So, this galactose molecule, it is getting converted to two things, UDP-galactose and glucose 1 phosphate. UDP-galactose can be converted to UDP glucose, and this can go to form glycogen, part of carbohydrate, again metabolism, right, metabolism of glucose, and it can be converted to glucose 1 phosphate, which is eventually converted to glucose 6 phosphate, and that can also go and take part in other metabolic pathways of glucose, right. However, you should know that galactose is not an essential. Why it is not essential? Because this enzyme UDP-galactose 4 epimerase is a reversible enzyme. It means even if there is no galactose to start with, so this galactose 1 phosphate is produced from galactokinase. So, galactose upon being acted by galactokinase forms galactose 1 phosphate, right, and this by galactose 1 phosphate uridyl transferase GALT is getting converted to UDP galactose. So, if this whole pathway is absent, how? There is less galactose in diet. Suppose there is low galactose, will the body have no UDP galactose? No, it will have. Why? Because we can always get UDP galactose from UDP glucose. The reverse pathway is very much feasible. Therefore, galactose is not an essential nutrient in diet, right. Only and only what we need this an action of this enzyme, the galactose 4 epimerase, right, and this requires NAD plus, right. So, galactose is not essential in diet. So, if you are facing an explain why type of question that galactose is not essential in diet, this is the answer, right, and there can be multiple choice type question MCQs that can be formulated from this section, right. There is an alternative pathway, very minor. So, let us deal with it now, that after 4 to 5 years of age, minded these all enzymes are produced in utero while the baby is in uterus, right. So, galactose metabolism starts because they are getting carbohydrate source from mother, nutrition via placenta, right. But so these all things can happen inside the uterus at birth. So, if these enzymes are deficient, the baby will have problems. We will be discussing very soon. But this alternate pathway happens after 4 to 5 years of age, when the baby has grown up a little. That is the galactose 1 phosphate pyrophosphorylase. This is one enzyme, a very minor enzyme. What will it do? It will directly produce UDP galactose, which can be converted to UDP glucose, right. Just know this for MCQ sake, that galactose 1 phosphate pyrophosphorylase can directly produce UDP galactose, okay, on which it will act on galactose 1 phosphate, of course, because whenever you are naming any enzyme, it is named after the substrate. So, galactose 1 phosphate. So, when galactose is there, galactose 1 phosphate pyrophosphorylase will directly produce UDP galactose. And why it is important? It is because if the baby is having deficiency of the previous enzymes, after he grows up to a certain age, the symptoms will be lessened. Anyway, this is a very minor reaction, but you might find a question here and there. But for all practical purposes, even if you do not remember the action of this enzyme, you are well and good. You have to remember the action of other enzymes, which if deficient produces a very important disease that is known as Galactosemia, right. So, considering the disorders of galactose metabolism, we move to the next part of the lecture, which is regarding the disorders. This is Galactosemia, and the most important variety of Galactosemia is classical Galactosemia. There may be many types of Galactosemia that we will also be touching very soon. This is an inborn error of metabolism. As I told you, the enzyme that is deficient is galactose 1 phosphate uridyl transferase, or GALT. Okay. So, if this enzyme is deficient, what will happen, and the incidence is 1 in around 35,000 birth. If this enzyme is deficient, galactose cannot be metabolized and there will be many problems. It is a rare congenital disease and it is inherited in autosomal recessive order. So, the rate of expression is actually very low, but still this is an inborn error of metabolism, means the baby is born with a disorder where one metabolic enzyme is absent and it is leading to expression of a clinical disease with multiple symptoms right from the birth. Okay. So, what will happen? You can easily guess. So, galactose 1 phosphate will accumulate in the liver. Who acts on galactose 1 phosphate? Galactose 1 phosphate uridyl transferase, this enzyme GALT, which is actually deficient. We are talking about GALT deficiency. So, if GALT cannot act on galactose 1 phosphate, there will be accumulation of galactose 1 phosphate, because the enzyme fructokinase is also, galactokinase is one way. Okay. So, once galactose 1 phosphate is formed, it cannot be converted back to galactose. After 4 to 5 years, I discussed there is a minor enzyme galactose 1 phosphate pyrophosphorylase that can help in conversion of this to UDP galactose, but till the baby grows up, right from the day of birth to 4 to 5 years, the action of GALT is extremely essential, and if it is deficient, there will be excess accumulation of galactose 1 phosphate, and this enzyme will inhibit galactokinase as well as glycogen phosphorylase. You remember what is the role of glycogen phosphorylase? Glycogen phosphorylase is an enzyme of glycogen breakdown, glycogenolysis, right? It helps in production of glucose from glycogen. So, if glycogen phosphorylase is inhibited, there will be less and less glucose, and that will lead to hypoglycemia. And since it is a disease of neonates or newborn baby, it leads to neonatal hypoglycemia, and galactose cannot be converted to glucose. Why we are relying on galactose to form glucose? The newborn babies are only and only dependent on mother's milk, right? There is no other source of glucose. You and I can simply drink multiple energy sources and we are getting ready-made glucose to start with. The newborn babies don't. Only mother's milk, that is lactose, is getting converted to glucose and galactose, and mainly whatever glucose is being converted, and galactose, it is also secreted directly from milk. Galactose is the primary source or food source in newborn babies. Okay. And this galactose, if it cannot be converted to glucose, the amount of glucose that is produced from breakdown of lactose is insufficient. We need galactose as well to be converted to glucose. I told you how it can be done. It can be converted to UDP glucose, it can be converted to glucose 1 phosphate, then glucose 6 phosphate. So, we need glucose metabolism to happen so that the newborn babies get energy from mother's milk, and if this does not happen, galactose 1 phosphate is accumulated. It is not only it is accumulating, it is inhibiting the formation of glucose inside the body, because all the substances that are present in the body from mother, suppose what will happen, we will discuss this in diabetes regulation of blood glucose. When we are starving, okay, when we do not have any food source, that is diet, our body actually synthesizes glucose by the process of neoglucogenesis. We are relying on that. And if in between meals, whenever the baby is having meal, mother's milk, it is getting converted from glucose, from lactose to glucose, some amount of glucose is going, galactose is not being converted to glucose, and in between meals it is relying on production of glucose from other sources, that is known as glycogenolysis and neoglucogenesis. So, stored glycogen will be broken down and other substances will be converted to glucose. If glycogenolysis is hampered, there will be hypoglycemia, and since it happens in neonates, it is neonatal hypoglycemia. Remember again and explain why type of question, if you see why there is neonatal hypoglycemia in galactosemia, this is the answer. Galactose 1 phosphate accumulates and it inhibits glycogen phosphorylase. There is inhibition of glycogenolysis, and this leads to neonatal hypoglycemia. So, increased galactose level increase during insulin secretion, which lowers the blood glucose level. So, what is this? What is happening? So, when galactose is increased, right, it leads to the liver, the pancreas, the beta cells of pancreas senses a monosaccharide, right. It triggers release of insulin. What is the role of insulin? It lowers the blood glucose. It is not a problem in normal individual, because in normal individual, even if the blood glucose is low, whenever we need, we can produce glucose from various sources. But in this case, not only is it preventing production of glucose from glycogen, it is also reducing glucose by triggering insulin release. So, everything is actually acting together to form neonatal hypoglycemia, right. So, what is the problem? Excess galactose will now remain in the circulation. Since galactose 1 phosphate is accumulated, ultimately there will be no requirement of converting galactose to galactose 1 phosphate. So, suppose some pathway A is getting converted to B, it is converting to C, I told you in feedback control. So, if something is blocked over here, the whole reaction will be blocked, right. There will be no more requirement of B to be converted to C, because this pathway is faulty, this conversion pathway is faulty. Again, this is also not acting. So, ultimately A will be accumulated, some B will be accumulated, right. So, excess galactose, if it is accumulated in the system, it actually is excreted in urine also. So, if we test blood, there will be high galactose level, that is known as galactosemia, right. If there is excess galactose in urine, this is known as galactoseuria, right. What happens? Bilirubin uptake is also less because the liver is at fault. Why? Because excess galactose 1 phosphate gets accumulated in the liver. It leads to a condition that is known as hepatomegaly. Megaly means growing of liver, right. So, hepatomegaly happens, there is liver is at an assault. So, the cells are filled with galactose 1 phosphate and the liver cannot function properly, and that is the reason why bilirubin uptake is less, bilirubin conjugation is reduced and unconjugated bilirubin is increased in the system. Basically, liver cells helps in conjugation of bilirubin. If liver cells are not functioning properly, again there will be jaundice, in this case unconjugated hyperbilirubinemia. Why the liver cells are not functioning properly? Because of accumulation of excess galactose 1 phosphate. Why it is getting accumulated? Due to loss of action of galactose 1 phosphate uridyl transferase, right. You get my point, right. So, enlargement of liver, jaundice and severe mental retardation, this is very important. Not only brain, liver, there is also mental retardation, because the food for brain is glucose. If the neonatal brain is subjected to hyperglycemia, brain cannot develop properly, right. So, there will be severe mental retardation, and this is the reason why galactosemia needs to be diagnosed early and it should be treated very early. So, just to look at what are the features or acute symptoms which a baby cannot complain because it is crying, okay, all the time. So, one of the reason of intractable crying of baby, I told lactose intolerance is one, this is one. So, swelling of the brain, edema, since they cannot digest it, the baby will refuse to feed. So, rejection of breast milk, a very important symptom of galactosemia, right. They will constant vomit. Jaundice, I explained to you why jaundice is happening. Pressure around the brain, our brain skull is in a bony cage. Suppose we have got trauma over here, something hits over here, if it swells up, there is no problem, it can swell because the bone support is inside, the outer side is free. But in case of brain, the organ is inside, if it swells up, there will be pressure that is being exerted by the skull because it cannot expand, that leads to increased CSF pressure, right. Again, there will be kidney issues. Why? Again, due to deposition of multiple byproduct, there will be excess unconjugated bilirubin, there will be problems of excretion of galactose. Why? See, kidney always try to reabsorb galactose, but if it is in too much excess, it has to be filtered and that leads to galactoseuria, right. So, liver failure, and this leads to a state of compromised immune scenario because the body is not getting enough nutrition, right. and these are the chronic symptoms. I will explain why there is cataract, feeding deficiency and growth issues because of malnutrition. I will very soon explain why there is cataract. Now, this galactose is getting converted to its reduced form. It is known as galactose is an aldehyde, okay, CHO. It is getting converted to a compound known as galaxitol or dalcitol, okay, with the help of the enzyme aldose reductase. This dalcitol or galaxitol once formed, it cannot escape the lens cells. This whole thing is happening inside the lens, okay. Galaxitol is osmotically very active. It attracts water molecules. When water molecule imbibes inside the cell, there is change of refractive index and the lens becomes hazy, okay. Moreover, this galaxitol also contributes to the injury of lens protein. So, this whole thing leads to an opacity in the lens in the form of cataract. Now, this was the major deficiency or major concern in galactose metabolism, that is the deficiency of galactose 1 phosphate uridyl transferase. However, the disease can also happen if there are deficiency of other enzymes. See, for example, deficiency of galactokinase. What will happen again? Excess galactose will accumulate, it cannot be converted to galactose 1 phosphate, galactose will be accumulated in the blood, they will be excreted in urine, but cataract is rare. Galactose can be converted to galaxitol definitely, right, but there is no galactose 1 phosphate. So, galactose 1 phosphate is not deposited in the liver and kidney. So, there will be absence of hepatic and renal complication. Cataract is also very rare, but still it can happen because of the aldose reductase enzyme. See, aldose reductase is converting galactose to galaxitol or dalcitol. So, considering the types of galactose or galactosemia, the main or the classical one is due to deficiency of galactose 1 phosphate uridyl transferase, that is considered as type 1. The symptoms more or less same, it is most severe in case of type 1 or classical, for the other case it is milder. So, for galactokinase deficiency it is milder, as I told you. It can also happen if there is deficiency of galactose epimerase. So, these are the genes. GLK1 is galactokinase, it codes for galactokinase. This is galactose 1 phosphate uridyl transferase, this is galactose epimerase, this is galactose mutase, which is actually triggering the conversion of alpha D galactose to beta D galactose. So, those are milder variety, but main is GALT deficiency. So, how we can diagnose? Diagnosis can be done in utero from drawing samples from the baby when even if he is not born, those are known as chorionic villus sampling, CVS, or amniotic fluid sample, and gene mutation analysis is done, especially the GALT gene. If GALT gene is deficient, we can easily predict that the baby is going to have galactosemia. But the treatment is actually very simple. We need to replace mother's milk, we need to remove galactose and lactose from diet, because lactose will again lead to production of galactose, and we use special formula diet for the baby. So, the key points to remember from this lecture is major source of galactose is lactose, that is obtained from milk and milk products. UDPgalactose is the active donor during any galactose synthetic reaction. Classical galactosemia occurs due to deficiency of enzyme galactose 1 phosphate uridyl transferase. It is one of the important causes of congenital cataract, and it is done by clinical evaluation of GALT enzyme and GALT mutation analysis, and treatment is done by removal of galactose and lactose from the diet. So, these are the references for this class, and I thank you for your kind attention.